Namaste to all. Welcome to the new session on drug discovery and drug designing. So in this session, we are going to see about the structural bioinformatics. So if you see the structural bioinformatics, it deals with many things. But starting with that, why we require this? The main reason why we require is because from the protein structure, when we are making the 3D model of that, that is having the like a template of that and unknown sequences there, based on the sequence alignment, what we can have is in the same order we can have the beta pleated sheets and the alpha helix and beta pleated sheets combined together to make a model in that. This model is actually called as homology model because in that the domains which are available here, based on the domains, what we can do is we can go for the drug designing or drug docking, you can say. That is the docking mechanism by which the target can be identified and that particular target can be highlighted or hit with our particular samples what we are expecting. So in this particular session, we are going to discuss the same. So what is meant by homology modeling? Homology modeling is also known as comparative homology modeling. That is comparison, means you are comparing the existing data, that is the unknown data, with your known data. So that is what is the actual thing is. So comparative homology modeling of protein is the technique which allows to construct an unknown atomic resolution model of the target protein from where its amino acid sequence and an experimental 3D structure of the related homologous protein. So that is acting like a template. See, for example, you are having already the template or a model of that particular 3D structure of a known sequence, that is the amino acid sequence is there. Similar to that, some sequence is there. Based on that, the structure is available. So that becomes as your template. And using that as a template, the known sequence, that is the unknown sequence, what you have got, what you can do is you can make a hit, and similar manner you can also construct the 3D structure. So this is what is being done with the unknown atomic resolution model of the target protein. So in the homology modeling, the basic thing is prediction of the 3D structure. So first thing is we have to understand that here we are making a 3D structure which we can rotate it and we can see that in which angle it is present, torsion it is present, and we can give based on the phi-psi angles and we can give it. So the prediction of the three-dimensional structure of a given protein sequence, that is the target protein from the amino acid sequence of a homologous template protein for which an X-ray or NMR structure is available based on an alignment to one or more protein structures. So if you have already done the sequencing of that particular structure, or you are having a protein molecule in which you have studied the X-ray structure or the NMR structure, based on that structure, this structure, the target protein structure, what you can do is using the computer softwares, you can predict the 3D structure saying that, okay, this is the angle in which it is present, in the torsion it is present, like that you can make it, and almost it will be similar with the correlating data of the X-ray or histography or NMR structures. So if the similarity between the target sequence and the template sequence is detected, then what we can do is the structural similarity can be assumed. So if, for example, the target sequence is our sequence, which we have found out, the new one, template sequence is already existing data. So based on that data, you are just comparing the target sequence, and the structural similarity wise, what you are doing is you are creating a new structure for this target sequence. So in general, 30% sequence identity is required to generate an useful model. So 30% is required. That is, similarity has to be there in the sequences. Based on that, the structure can be made here. So if you see in detail here, what is the sequence similarity and the structural similarity? That is, as long as the length of the two sequences and the percentage of the identical residues fall in the region marked as safe, the two sequences are practically guaranteed to adopt a similar structure. So the basic thing is, say for example, this is your identity. Okay, so this is the identical sequence, or you can say the template part. The similarity part, what is happening is this is like a similarity. So almost there is some similarity is there between these two sequences. So there are more similarities are there in this particular region when compared to this. So this is how the comparison software actually works and gives you the data. So here you can see that how much the percentage of similarity is there, based on which you can say. So till 30%, you can say that assignment of function or the protein fold, focus or mutagenesis experiments, then finding the binding or active sites by 30 motif searching, then NMR structure, that is the refinement. And from 30 to 60%, it is depending upon the properties that do not occur in the template. So the do not occur in the template means in the template some different properties will be there. Like electrostatics or cavity volumes, how much the depth is and everything, and the protein engineering, that is the stability part, okay, and the molecular replacement, which has to be which has been studied in the X-ray crystallography, that will be highlighted here. Then from 60 to 100%, that is like maximum, the comparison will be taken up with the experimental data, that is based on the X-ray crystallography or NMR studies. So the comparable to the medium resolution X-ray or NMR structures, then structure-based design and understanding the substrate specificity. Substrate specificity means how much the substrate specificity is there, how the angle is there for that, so that the active site is present. So all these things will be studied here. So as we have said the concept of the homology model, what we are doing here is we are taking our unknown sequence. With that, we are having a template. This template, based on that, what we are doing is the sequence is already there with our unknown sequence and the template sequence. So that we are comparing. When we are comparing, if almost like 30% similarity is there, more than that is there, what we can do is taking this as a template, we can create a new structure for that unknown structure, that is the unknown or you can say the target sequence. So what we get a 3D structure model like this, that we can present it, that this is the model of our unknown structure, what we have, unknown sequence or the target sequence, what we have found here. So the structure prediction by the homology modeling, if you see how to identify this. So first is based on this flowchart, you can identify, that is the identify the related structures. So related structure, that becomes as our template. Select that particular templates, then align the target sequence with our template structure. So this is how the sequence alignment is being taken place. Once the sequence alignment is done, then what we are going to do is build a model for the target using the information from the template structure. So in the same order, we are taking up the data and we are creating the model now. So evaluate the model. If the model looks okay, that is by seeing that it is looking with the pleated sheets and the alpha helixes and the connection, everything is okay, then what we can do is we can go with the yes and end the sequence. If it is not, then we have to again search for our new template, and once the template is being aligned with our particular data, that is the target sequence, and the sequence and the structure looks good, then we can take it up further for further studies. So let's take an example here. So to the known structure of sequence A, which is having 150 amino acid long sequence, what we are doing is first of all, we are going to compare the sequence A to all the sequences, because existingly in the PDB, that is a protein data bank, we are having all the sequences. So in the protein data bank with the known structures, we are comparing our sequence A. So using the BLAST. After doing this, if the sequence B, that is the 300 amino acid long sequence, is containing a region of 150 amino acids, which are similar to our sequence A with 50% identical residues. So we have said that more than 30% has to be there. Now there is a sequence B, which is 300 amino acids. In that, this 150 amino acids are also falling and they are having a similarity of nearly or identical of nearly like 50% identical residues. Now what we are going to do, as this match alignment takes place, it clearly falls on the safe zone. Safe zone means everything is comprising. So we have seen that 30 to 60% is the range in which it has to be similar to the X-ray crystallographic studies and other protein depth and other things, we are seeing extra points like electrostatic and value points and everything we are seeing here. So what we are doing here, we can simply take the known structure of the sequence B as our template, cut the fragment corresponding to the aligned region. So where it is having, in that region, we are cutting it and we are taking only the region where it is having the similarity. Then mutate those amino acids that differ between the sequences A and B. Okay, so where there is some differences, we are just mutating it. So what we get, finally arrive at our model for the structure A. So for this, now we get the structure A, which is similar to the structure B, almost we can say. So the structure A is called as the target and is of course not known at the time of modeling. So till the modeling gets over, we will not know that whether the structure is going to come or not, because we have to see the simulations and similarities. Based on that only we can work. So let's see about the history where it all started. The first homology modeling studies were doing using the wire and plastic models of bonds and atoms as early as the 1960s. The model were constructed by taking the coordinates of a known protein structure and modified by hand for those amino acids that did not match the structure. So in those days, since this modeling, everything was not there, so they used the wire and plastic models like balls and all, and then they made the models and they presented it. So in 1969, David Phillips, Brown and co-workers published the first paper regarding the homology modeling, and the modeled alpha-lactal albumin based on the structure of hen egg white lysozyme. So that was the first study which they did. And the sequence identity between the two proteins were nearly 39%. So at that time they made it manually and then they analyzed it. So that was nearly like 39% similarities were there. So what are the different steps which are involved in the homology modeling? There are totally 7 steps which are involved here. So the first step is called as template recognition and initial alignment. Second step is called as alignment correction. Third is called as backbone generation. Fourth is called as loop modeling. And fifth is called as side chain modeling. And sixth is model optimization. And seventh is model validation. So all these things are actually present here. So we will see one by one in each. So first, going with the template recognition and initial alignment. So what we are doing is the template is there. So template is already existing in the data bank. So template recognition and selection involves searching the protein data bank for the homologous protein with determined structures. Determined structures means already existing structures are there, already the sequences are there, the homologous sequences are there, that we are taking and we are collecting the data. Search can be performed using the simple sequence alignment program such as BLAST or FASTA, as the percentage identity between the target sequence and the possible template is high enough in the safe zone to be detected with these programs. So that is what is present here. To obtain a list of hits, that is how many matching is there, that is the modeling templates and the corresponding alignments, the program compares the query sequence, that is what we have provided, to all the sequences of the known structure. So what are the different sequences are there? For that corresponding the structures have been placed. With that, the sequence will be compared. So what happens in the data bank, that is the protein data bank, when they compare with all the sequences which are present, we will get our required data which is having the similarity. So there are 2 matrices which are followed here. The first thing is a residue exchange matrix and second is alignment matrix, which is used almost similar to the dot matrix, you can say. So here, if you see, it is how the matrices looks like. So this is our first protein sequence and this is our second protein sequence, which is being kept on the top as well as at side. And the second one is for alignment part. So in this, how much the alignment is happening or how much the sequence similarities are there, that will be studied. Sequence A, sequence B similarities, how it is there, and everything will be studied based on the particular matrix scores. The second thing is alignment correction. So alignment correction, what we are doing is Sometimes it may be different to align two sequences in a region where the percentage sequence identity is very low. So if it is below 30%, what will happen? But similarities are there. Below 30% score is not there, then what we will do? So for those cases, we have to go for alignment correction now. So one can then use other sequences for homologous protein to find a solution. So what we have to do? We have to take up another sequence and then compare it with our sequence, and then we have to find out for the homologous proteins here. So you can see a sequence A and sequence B are there. Sequence A is having LTL, TL, TLT, and the sequence B is having YA, 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 Y. Okay. So how much the similarities is there that we are going to align and we are going to check. So for that, what we are taking is, for example, to align the sequence TL, 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 T with YA, 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 Y, which is nearly impossible because in that T is also not there. In below, there is no L also. So what happens? It is not going to match. But now what we are going to do is, we are going to take the third sequence. So the third sequence is TY, 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 T. So this is, you can say that in this T is there, in this Y is there. Okay. So we can align both the sequences now. So keep the third sequence in the middle, first sequence on the top and second sequence in the below. So then we are going to compare it. So this is how we are going to take it and we are going to do the analysis. So here, this is our template and this is our model 1 and model 2. So if you see carefully, I think the visibility of the second sequence is not that much, but you can see here what is happening. So you can see here the first model in this, you are having the phenylalanine, asparagine, valine, cysteine, arginine, alanine, proline, and there is a big gap is there. Then you are having glutamate and alanine and isoleucine. But in top, if you see the number 13, number 10, number 9, number 8, okay, and then number 6, then number 3 and number 2. So in that only we are going to have a change. So for that, what we are doing, we are taking another model and then we are again comparing it. So what happened? Few regions have been covered up and or it is going to be nearby. So 2 is correct because it leads to a small gap compared to the huge hole associated with the alignment 1. So if you see here, here there is a big gap which is occurring here because there are so many things are there which is not matching. And another thing is the sequence 2 which is there in the below, it is having a lesser gap only when compared to the upper one. So when we are aligning this like this, so what we can get is we can find that few of them. Okay, so this is the correct alignment which is taking place. So based on that, we can correct it and we can identify it. Okay, so the known structures based on that model and everything has been provided here. So backbone generation. So backbone generation is very much important here. So when the alignment is correct, the backbone of the target can be created. The coordinates of the template backbone are copied to the target. And when the residues are identical, the side coordinates also copied. So what happened? Everything, the target wise, the backbone, everything is being set and then it is being aligned now. So loop modeling. So loop modeling, if you see after the sequence alignment, there are often regions created by insertions and deletions that lead to the gap in alignment. So these gaps are modeled by loop modeling, which is less accurate. So what happened? Less accurate means what happens? It will not work. So we have to check for the data. So the currently 2 main techniques are being used for this approach for this particular problem. That is the database searching method and ab initio method. So the database searching method, how we are doing is, this involves finding the loops from the known protein structures and superimposing them onto the 2 stemmed regions. That is the main chain mostly of the target. So what happens? We are comparing it. Okay, if it is any changes are there or any loop changes there or loop is having some issues or anything or less accurate. So for that, we are comparing it with our known protein structures. So some specialized programs are there like Freed or Coda that can be used here. Then ab initio method, if you see, that depends upon they generate the random loops and searches for one that has a reasonably low energy. So in which very low energy is there, that is going to be identified, and also the psi and phi angles in the allowable region in the Ramachandran plot is there or not. So Ramachandran plot is very much important in this particular scenario to create the 3D modeling. Okay. So we will see this ab initio in a detail in the next session. So how it works. So in this, the red loop is modeled with the green residue as an anchor residue. The insertion of the second residue results in a longer loop now. So this is how the loop is being corrected and it is being modified. Then side chain modeling. So in the side chain modeling, what we are doing is, this is important in evaluating the protein ligand interactions at the active sites. So at the active sites, what is happening? The protein is there, ligands are there, that which is protruding out and it is reacting, how it is taking place, and also for the protein-protein interaction. So for these two proteins, how they are interacting at the contact interface. So a side chain can be built by searching every possible conformation for every torsion angle of the side chain to select that one that has the lowest interaction energy in the neighboring atom. So if, for example, the side arms are very huge, okay, the bulky chains are there, like you know that certain proteins, amino acids are there, in which the bulky amino acids, if they join, what happens? They will be protruding out. So it will be very bulky, so it will be for joining or for making the helix properly, it will not be proper. So they have to be in such a order that the sulfur groups or the bulky chains should not be there. So the torsion angles and everything plays a very important role here, and also the lowest interaction energy here. Rotamer library can also be used, which is also which has all the favorable side chain torsions angles extracted from the known protein crystal structure. So from the X-ray crystallographic studies, from that we can know that which is the correct angle in which we can take it, or which particular amino acids will be suitable. So the side chains, so that based on that we can take it here. So model optimization, once everything is there, the model optimization has to be taken place. So the energy minimization procedure of the entire model by adjusting the relative position of the atoms so that overall conformation of the molecules has the lowest possible energy potential. So we are reducing it and we are taking it so that the model can be used for various purposes. So the goal in this is to retrieve the steric collisions. Steric collisions means how the collision is taking place, the steric bonds, okay, so altering the overall structures. Then optimization can also be done by the molecular dynamics simulations, which moves the atoms towards the global minimum by applying various simulations, conditions like heating, cooling, considerable water molecules. So all these things are parameters, so by which they come closer, and thus having a better chance at finding the true structure. So by this, what happens is we get a true structure of that particular unknown sequence. So energy is equivalent to stretching energy plus bending energy plus torsion energy plus non-bonded interaction energy. So all these things plays a very important role. So angle bending is also there, bond stretching is also involved, non-bonded interaction is there. Like non-bonded means since they are not bonded, but the interaction will be there. They will be pulling each other closer. And torsion angles are there. That is, one will go like this, one will go up. So in which angle it is going. So that also plays an important role here. Then after finishing everything, model validation has to be taken place. So every homology model contains errors. There can be error because it is not from the same sequence. We are taking a template which we are aligning it and we are taking the minimum percentage of identity. That is, you can say like 30, more than 30% identical, then only we are taking it for the modeling purpose. So that is why error chances are always there. So the two main reasons for this is the percentage sequence identity between the template and the target. If you see, that is, if it is greater than 90%, the accuracy of the model can be compared to the crystallographically determined structures, and if less than 30%, large error occurs. Okay, so that is what is the difference. If greater than 90%, accuracy will be there. If lesser than 30%, errors can occur. So that is also there. So the number of errors in the template is being counted here. Then what we are going to see, about the phi-psi angles. So the final model has to be evaluated for checking the phi-psi angles or psi-phi angles. Then chiral bonds or chirality. Then bond length, how much the bond length is there. Then close contacts, what are the close contacts are there, or side chains or amino acids are there, or how it is interacting. Then stereochemical properties, how the property is there, how the torsion is there, how the angle is there. So everything will be analyzed. Then the modeling programs are being used here. Basically, they are modeler, Swiss model, Schollinger, or 3D jigsaw. So all these things are the softwares which are used for the homology modeling. And the successful model depends on the template selection. So template selection means it has to be a good template, which is having minimum, there is a minimum gaps and maximum matches, basically you can say. So algorithm used and the validation of the model. So all these things plays a very vital role for the homology modeling. So what is the advantages of this, of homology modeling? It can find the location of the alpha carbon of key residues inside the folded proteins. So we can find the alpha carbons, that is the most important thing. So how the distances between the alpha carbon and the beta carbon, you can say, or alpha carbon and the nitrogenous bases, how it is in the bases, that we can check here. So that is one part. It can help to guide the mutagenesis experiments or hypothesized structure function relationship. So in mutagenesis experiment, if any mutation has happened, how the structure looks like, so what is the role it will be, or how the domain will be. So everything can be understood from this particular structure analysis. And the position of the conserved regions of the protein surface can help identify the putative active sites and binding pockets and ligands. So if it is always conserved, that is from generation after generation, this particular region is conserved, then we can identify the putative active sites. Putative active sites means which always be active and which will be reacting with our substrates, basically. What is the disadvantages of this? The disadvantages of this is the homology models are unable to predict the conformation of insertions or deletions or side chain positions with a high level of accuracy. That is a very biggest difficulty here. What happens is, if anything has been added during the sequence, if anything has been inserted or deleted, that we cannot identify here. Another thing is the side chain position. That is, we have seen that side chain, we have to carry out the side chain, but the position where it is present and how much accuracy it is, that we cannot say here. The second part in this is it is not useful in modeling and ligand docking studies necessary for the drug designing and the development process. So this is useful for designing or for structuring, it is useful, but for docking studies, that is for designing a new drug so that the substrates can bind at the active side and can be reacting, for that this homology modeling studies is not so suitable here. And however, it may be useful for the same if the sequence identity with the template is greater than 70%. If it is more than 30%, it is not suitable for docking. But if it is more than 70%, then you can consider for the docking purpose or for the docking studies in the drug discovery. Next, going on with this, the important part is Ramachandran plot. So Ramachandran plot, it is being in a polypeptide chain. You know that the main chains are N and C alpha. That is, C is in the center. Next to that is NH3. That is N C alpha. And then you are having CC. That is C alpha and COOH. So C bonds. So N C alpha, C bonds. Relatively are free to rotate. So they can rotate in an angle, they can go up and they can rotate. So these rotations are represented by the torsion angles. The torsion angles means how much the distance is there. Based on that, the angle is being provided in phi or psi angles. Okay. So this is being given by, this is called as a Ramachandran plot or phi psi plot. Originally, it was developed in 1963 by a group of Indian scientists from Madras University, that is the biophysics department. They are called as G. N. Ramachandran, C. Ramakrishnan, and V. Sashishekaran. So these are the three scientists who are involved in the X-ray crystallographic studies of protein molecules in biophysics department of Madras University or University of Madras. So they only proposed this particular concept of Ramachandran plot, which is still now very much useful for the understanding of the structures. So this is a way to visualize the backbone dihedral angles, that is the 5 again the psi of the amino acids residues in protein structure. So how it can be used, how the Ramachandran plot can be used. One is to show in theory which values or conformation of the 5 psi angles are possible for the amino acid residue in a protein. So that is very much important. In theoretical aspect, you have to mention that, okay, this is the angle of the phi, this is the angle of the psi and of the particular amino acids in a particular protein. Based on that, the second thing is to show the empirical distribution of data points observed. So empirical distribution of the data points, that is where it is bending, where it is giving the angles, a single structure in the usage for structure validation or else in a database of many structures. So when they are compared with many structures also, you can take up the data and you can compare it. So this is how the Ramachandran plot looks like. So the first thing is the left-handed alpha helix. The second part is beta pleated sheets, and third is for the right-handed alpha helix. So all these things taking into care, you are going to get your 5 psi angles, and based on the 5 psi angles, the structure is being provided and the torsion angle is being provided for the particular amino acid sequence, and then relating to that particular angle only, this homology modeling is being performed. Okay, so with this, I hope that you have got an idea that how the homology modeling is being considered and how the homology modeling takes place in the 7-step process. So thank you and see you in the next session with more details about the drug discovery.